Hello dear students, I am Assistant Professor V.S.Aher, and in the last session of Microbiology part first, we have already started with the nucleic acid, in which we have studied what are nucleic acid and what are the types of nucleic acid, in which we have already studied about the DNA. In today's session, we are going to study about the RNA molecule, or it is also known as ribonucleic acid. Now, the RNA, it is a polymer of ribonucleotides, and most of the RNA, they are single stranded. It is long unbranched macromolecule which consists of nucleotides joined by 3' to 5' phosphodiester bond. Basically, they can form or they can fold upon themselves in region, and here the complementary base pairing is possible to form the secondary folded structure. In simple words, RNA is a polynucleotide chain, means different nucleotides comes together and join to each other with the help of phosphodiester bond, and it forms a polynucleotide chain which contains ribose sugar, phosphate group and nitrogen bases. The number of ribonucleotides in RNA, it is around few, for example 75 to many thousand, means near about 75 to many thousand nucleotides comes together and form a polynucleotide chain, and to these polynucleotide chain various ribose sugar, phosphate group and nitrogen bases attach to form a RNA molecule. Now if we consider about the structure of RNA structure is quite similar to the DNA. Both have the sugar and the phosphate polymers which contains the nitrogen bases. But though they have the similar structure, there are few of the differences present between the structure of RNA as well as DNA. Now there are three basic differences present in both of the structure, in which the first difference is the RNA contains a different monosaccharide residue. The sugar present inside the RNA molecule is slightly different than the DNA molecule. If we consist about the nitrogen bases, the nitrogen bases present inside the RNA, they are adenine, guanine, cytosine and uracil. Instead of uracil, in DNA thymine is present. So in DNA molecule, the nitrogen bases which are present, they are adenine, guanine, cytosine and thymine molecule. And the third important difference between DNA and RNA molecule is RNA is present in single stranded form, whereas DNA consists of double stranded structure. Here you can see the structure of RNA. It is a single stranded structure which consists of nitrogen bases like cytosine, guanine, adenine and uracil. In RNA, the purine to pyrimidine ratio is not equal. and RNA acts as a genetic material only in certain viruses. In other cells, RNA is not, or it is non-genetic material, and there are three types of RNA which plays a vital role in this inside the cell, which includes mRNA, r-RNA and t-RNA. We'll study all these three types of RNA in detail. First one is our t-RNA. Now the t-RNA, it is known as transfer RNA. The transfer RNA, it is the smallest of all the types of RNAs. It consists of near about 73 to 93 nucleotides, and it is well characterized and completely sequenced RNA. It is present near about 10 to 15% inside the cell, and its key role, or the important role of tRNA, is molecule in the protein synthesis that are transferred the amino acid from the cytoplasmic pole to the site of protein synthesis. As here, these RNA, it transport amino acid to the site of protein synthesis. It is known as transfer RNA, or it is also called as adapter molecule. It exhibits extensive inner chain of bonding which represent the cover-leaf structure. Its structure is slightly hairpin like model or clover leaf model. Second type of RNA is ribosomal RNA or r-RNA. Now the ribosomal RNA, it is also single stranded but may be variously folded and show complementary base pairing at the folds. It is present near about 75 to 8%, 80% inside the cell, and these ribosomes, they are present at the site of protein synthesis. And as compared to the tRNA, the ribosomal RNA or r-RNA is slightly greater in the size. And the third type of RNA is m-RNA, which is known as messenger RNA. Now the messenger RNA, it is linear molecule which does not contain any kind of folds. As it carries the message from protein synthesis so it is known as messenger RNA, or it carries the message for protein synthesis from nucleus, that is from DNA in the nucleus to the cytoplasm. As the message from the DNA to the cytoplasm is passed by this RNA, so it is known as messenger RNA. This messenger RNA is synthesized from the DNA molecule by the process of transcription. The size of this messenger RNA may vary, and in most of the cells near about 5 to 10% of the total cellular RNA, the mRNA is present. So now we will see the functions of different types of RNA. First we will see the function of mRNA. Now of mRNA, which is known as messenger RNA, its important function is transformation of genetic information from the genes or from DNA to the ribosomes for the protein synthesis. The main role of mRNA is the transformation of information from the DNA to the cytoplasm or the ribosome. And the second type of or the second subtype of RNA is hnRNA, which is also known as heterogeneous nuclear RNA. These heterogeneous nuclear RNA, it serves as a precursor for mRNA and other RNAs. The third type of RNA is ribosomal RNA, which is known as r-RNA. The ribosomal RNA or r-RNA, it provides the structural framework for ribosomes. A proper structure to the ribosome is provided with the help of ribosomal RNA. Then the role of transfer RNA or t-RNA is the transfer of amino acid to mRNA for the purpose of protein synthesis. Then there are some other subtype of RNAs like snRNA, which is known as small nuclear RNA. These snRNA, they are involved in the mRNA processing. mRNA, when it plays a role in protein synthesis, for the processing of this mRNA, the important role is played by the small nuclear RNA. The next type of RNA is snORNA. Now, snORNA, it stands for small nucleolar RNA. And these small nucleolar RNA, it plays a key role in processing of ribosomal RNA molecule. The role or the functions of ribosomal RNA molecule is processed with the help of snORNA. Then the next type of RNA is small cytoplasmic RNA. This type of RNA, it is present inside the cytoplasm and it involves in the selection of proteins for the export. Here it selects the protein molecule and then it transfers it to the other parts of the body. And the last type of RNA is TM messenger RNA, which is transfer messenger RNA. It is mostly present in bacteria. And these transfer messenger RNA plays an important role in the degradation of incorrect synthesized protein. Whenever a proper protein is not synthesized inside the cell, then such type of incorrect protein is degraded or break down with the help of transfer messenger RNA. So these are the types and functions of the different types of RNA. Now here are the nucleic acid and the protein synthesis processes. Now the nucleic acids are synthesized with the help of replication process, the process by which replica or the identical copies of DNA is made inside the cell during cell division. That process is known as replication. Then the process of synthesis of mRNA from the DNA molecule, it is known as transcription. And after the synthesis of mRNA, the process of synthesis of protein molecule with the help of mRNA, it is known as translation. So here is the central dogma. From DNA, there is synthesis of RNA molecule. This process is known as transcription. And from mRNA, there is synthesis of protein molecule, and this process is known as translation process. When a replica or a copy of DNA molecule takes place, then that process is known as replication process. So this was all about the nucleic acid and the types of nucleic acid, as well as we have also seen the structures of nucleic acid. Thank you.